coming. Thank you for the introduction. So yeah, my name is Noor Stern. I opened my lab here about less than 2 years ago. It's called Dentistry, and my lab is interested in understanding how organisms form their shapes of their organs throughout their early development. And we do that in two model organisms. One of them is the early fruit fly embryo that you will hear about today. The other one is the early mid development, embryonic development of the mouse. And we do that mainly by using and developing bioimage informatics tools to tease apart the complexity of live imaging data, as I'm going to show you today. So there's going to be 3 chapters. First one, I'm going to show you how we do whole embryo cell segmentation and tracking, and then I'm going to show you how we identify and map cell activities over time in the early fruit fly embryo. And then I'm going to tell you a little bit about an ongoing project, which is supposed to make all of these computations very accessible to everyone, experimental labs included. Okay, so, one of the most remarkable things about nature is this endless repertoire of morphologies that we see around us, and questions about the genetic patterning or molecular patterning of these activities and how cells perceive them and organize in time and space, carry out their activities that in turn deform organs and deform the entire embryo, have fascinated biologists for centuries, actually.
Forever, yeah.
Forever, right. And we're living in a great time to be doing this science because over the last 10, 20 years, microscopy reached the point that we can image whole embryos every few seconds at submicron resolution, which gives us literally direct view of everything that is happening in the embryo. But it's already very clear that without the help of computers to untangle, to mine this information, to visualize it for us so that us humans with the limited brains could actually perceive all of this complexity, we're pretty much stuck. So just to give you an analogy here, so think about the sequence analysis community. So ever since the early 70s, when people started to do sequencing, so geneticists did a very smart move. They made friends with computer scientists to develop for them algorithms for identifying open reading frames and pairwise sequence alignment, and then it was the BLAST family of sequences, and today we even have these deep learning versions that give you hypotheses directly from the data. Now, arguably, the movie of an entire developing organism is as complex and as dense information as the DNA that encodes it, but nevertheless, most of the information that we see there is simply remaining unnoticed. And so one of my lab's goals is to bridge this technological gap in what I'm going to show you today. Okay, so whole embryo cell segmentation and tracking, the first chapter. In case you're not familiar with that, this is called Drosophila melanogaster, aka the fruit fly. Probably one of the most, or not the most, used model for genetic and morphogenesis in biology. And one of the most studied processes is called gastrulation, more or less the first and biggest thing that happens in development, where we start from a monolayer epithelium and through coordinated cell activities, which are described, transforms into the 3 main germ layers, which are endoderm, ectoderm, and mesoderm. And these main cell activities are basically the changes in cell shapes, cell division, and the rearrangement of cells, exchange of neighbors. Now, it's obvious that, like for 20 years or more, we had a magnificent breakthroughs in understanding the genetic patterning that guides the cell on how to do these activities, and in the cell biology that shows us the mechanisms that interpret the signals and execute them into mechanical forces. However, we still have huge gaps in our understanding of how from the cell level to the whole tissue or the whole organism level, this information is propagated. For example, what are the impacts of one tissue developing on its neighbors? This is a very hard question that people are still very much struggling with.
And what's the time frame of that movie? I mean,
this movie?
Yeah.
Well, I would say about an hour-ish.
I see.
It starts about 3 hours after egg laying, and it's about 1 hour long.
My God, that's incredible.
That's like
That's 1 hour.
That's
Yes,
that's
what a dramatic change in an hour.
in 20 seconds a cell will stop overlapping with itself from the next time point.
Yeah.
So I'm saying like 20 seconds will come here. Yeah, it's remarkable.
Huh. Thank you.
Right. So clearly, in order to do this research, we cannot just look at the tissue because it's way too complicated for us. So what we need is to be able to extract from the tissue the shapes of the cells over time, which is what we call segmentation. And we need to also know how to track them over time, to link the same cell within different time points. Now, of course, there are thousands of papers that show how to do that. However, until the work that you're going to see now, none of them was able to segment and track all the cells in the entire embryo over any period of time. Okay. So with that in mind, we decided to develop that algorithm and then to develop also some mining and visualization in order to simplify this complex information for human eyes. Okay. Before we start with the algorithmic, you need to understand how we get this data because, you know, for the last 20 years, we had confocals that basically dominated the biological microscopy, and people did and still do fantastic work with that. But if you want to go to whole organism imaging, confocal will penetrate more or less 50, 60 microns into the tissue. It will take plenty of time to do that because it's a point scanner. At each time point, it's taking only one pixel. And it will also bleach your tissue. So you can, by definition, not do anything whole fly embryo with confocal. For that, we're using line sheet microscopy, a technology that, let's say, came back to life about 10 years ago. And the idea here is a bit different. So we have here two illuminating beams here that create this laser line. This laser line fluctuates like 70K hertz or something like that, that creates the illusion that you have a sheet here. So this is why we call it a light sheet. We're line, it's a line, but it's a very fast one, so we call it a light sheet. And then think about like a mid slice that you pass the tissue through that, and every time it shines an entire plane from the tissue, and the information from the fluorophore is then being collected by two orthogonal lasers from the two pink ones, and in a matter of 3, 4, 5 seconds, you can take the entire fruit fly at submicron resolution. It's a remarkable thing. Yeah, it doesn't look at all like a microscope. This is our light sheet, and it's a really weird machine. So just to give a sense of what a light sheet movie looks like, this is a single time point of a fruit fly embryo that is expressing an endogenous membrane marker. This is actually slower than the time of acquisition. The acquisition will take much faster than that. And of course, we can do that very fast over time. So here we have an example, the same embryo, we're just showing the movie. We took here every 40 seconds. We don't need more than that. Snapshot every 40 seconds.
The frame rate is 1 every 40 seconds.
Right, it's a 3D, once in every 40 seconds.
Okay, good.
And it's not only one 3D, it's multiple channels of 3D. So this is the same fly embryo. We also did the nuclear channel, which is a different fluorophore. And so when we look at them together, we have all of this information. We can do up to 4 channels at the same time. Right, and you can penetrate the entire embryo. You can see everything basically.
How big are those files?
Files?
Yeah, what's the file?
Before, after reconstruction.
Well, just any way you want to answer. Just the raw data.
We're easily generating terabytes a day.
Terabytes a day.
Yeah, but we throw most of that because you're collecting 2 cameras at the same time.
Yeah.
Right, you don't need that. We fuse that. And then when we fuse that, it becomes half. And then you can shift it to 8 bits, and you don't need the full resolution. So we're turning that, let's say that each one of these movies is like 100, 200 gigs. Let's say like a gig or 2 for time point, if you want a good quality. Yeah. H5 is very helpful. You can compress it very well.
Thank you.
So, okay, so we have the data. Fantastic. Now we can go to do segmentation. So, the strategy that we took is called watershed propagation, and it goes like this. Well, first of all, we're focusing here in this segmentation only on the 2-dimensional surface that is located about 5 microns under the apical surface. The reason for that, well, technologically, of course, it was easier. And also, 5 microns is where most of the mechanical machinery of these cells is located. So this is where it's interesting to look at. So what we do is we take the surface, and first thing we do is we segment the first time point manually or with Fiji, Imaris, whatever you want. as long as you make sure that you did a perfect job. Now, for the second time point, you can this is segmentation of the first time point. For the second time point, you can, in principle, do the same thing manually, but it will take you like your entire PhD to do that for the entire movie. And also, if you manage to do that, you still didn't solve the problem of how to link the cell, how to track the cells over time. And so, watershed propagation offers us a way to use one stone to kill two birds. And the way it works is this. So you overlay, let's say that we're overlaying the first time point on the second, the green and red here. Obviously, since the embryo is 40 seconds older in the red, the cells are not overlapping. But since it's only 40 seconds, what we can do is apply a deformation algorithm that will identify perfectly the matches at the single pixel level between them. So now that we have the displacement map, what we can do is we can just apply it on the segmentation of the first time point to get the second time point. And that solves both problems, because now we know the segmentation of the second time point, and we also copy the identities of the cells. And so, like a mathematical induction, you can continue from second to third, third to fourth, and so on until time point 1000. Okay, so watershed propagation, this strategy is not our invention. It's about 20 years old. The only problem is that it was designed for simple, flat, two-dimensional images and not for these curved and ever-changing surfaces of the embryo. So we have a problem here. So the way to bypass that is we use what we call a Mercator projection. So Mercator is what you do when you want to take a complex surface in a 3D space and just flatten it. It's exactly what you do when you take the globe and flatten it. The only problem is that when you do that, the Antarctica and the Northern Pole stretch actually to infinity at the top and the bottom lines. And so we don't want our cells to stretch like this because we won't be able to handle them. So what we do is we simply apply that to 4 different regions in the embryo and somehow regularizing how much they're stretching. By the way, this algorithm was written by Itza Himskirk, which is, I think he's also somehow social. He's here basically in Michigan, right? And so once we do that, we can do that also for the second time point, and then we can apply the traditional version of watershed propagation between the 4 regions. We then do inverse Mercator projection, and we end up in the regular embryo, the 3D embryo of the second time point, right? Okay, so we're talking about thousands of cells for hundreds of time points, and we're flattening the embryo and we're breaking it into 4 parts and putting it back. How accurate can this thing be? So in order to evaluate that, we took a random sample of 15,500 cells, and we checked that each one of them is being segmented and tracked properly between the 2 time points. And so we found that about 98% of them were done perfectly well. And out of the remaining 2%, 3/4 were in regions like here, where the cell, is just about to invaginate to the embryo, and so the membrane is kind of overlapping with the plane that we're analyzing. So it's almost like there is no definition of a proper answer here. So it's an excusable error, you can say that. Now, how does it compare to other algorithms? It's a bit difficult to compare because algorithms today don't do a 2-dimensional fold in a 3D surface. They either do 2D or 3D, but I think that the best today is about 50% errors or 50% correct tracks over 62 time points. And we're doing about 98% over 200 time points. So in our category, we can say at least we are very competitive in this. We're very happy about this algorithm. Okay, so just to give you a sense of what the results look like, you're looking at the same algorithm same embryo that you saw before, 3 different views, and when I would click, you could see all the segmented cells over time. And if you're interested to see the tracks of the cells, how they move, in this movie, you'll see the centroid of every cell randomly color-coded moving over time. Okay. By the way, feel free to like if you have questions or whatever, just stop in a minute. Okay, great. So now we have all the cells segmented and tracked, and we have the computerized data, and we can go to analyze it. So one of the most important cell activities during development is every process is of course cell division, and yeah, just to give you a sense of what it looks like in our perspective. And I don't suppose that you managed to see that, but in the movie you just saw, you had 1,108 cell division events that were distributed over the embryo and at different time points. And obviously, great, we have the cells segmented, but it's still too complicated for us to see that. So we need some methods to simplify it for us. And so we came up with something extremely simple, which is we call the blastoderm projection. Since we have the ability to track the cells back and forth, what we do is we simply take every mother cell to be on the first time point of the embryo. First time because this is the most simple and no faults, and it's easier, and people know, Drosophilists know this, what it looks like. And we color code it by the time in which this cell is going to divide in the future. So blue cells are going to divide in about half an hour, red cells in about an hour, and white cells are just not dividing during our movie. And of course, you can apply the same trick with the Mercator projection, which allows you to see the entire pattern in front of you. It's very symmetric. It's just beautiful to see this biology in front of us. So how accurate is this information? The only reference that we had for mapping these cell divisions was from a paper by Victoria Bowe from 1989. She's today an emeritus in the University of Washington, I think. And she worked for quite a few years to map the 25, I think, mitotic domains of the early fruit fly, in a paper that became everyone in the field knows this paper. And we compared our results to Victoria's results, our domains to her domains, and we found just simply 100% agreement. And if you ask me, this is just an indication of how phenomenal her work was. I mean, we're using computers, she did it with her brain. So, but nevertheless, we're using computers, and there are some advantages for that. And of course, the advantages are that we're working at single cell resolution and not in manually sketched domains. We know when the cell divides in plus minus 15 seconds. We need only one embryo to do that. We had only one embryo back then. So, and the last thing is that once you have the data, you just need to click the enter and it's being done for you. So that's a big advantage. And so this was a useful trick. And we said, okay, so why don't we just, build an atlas of morphogenetic fate maps, if you will, of any cell activity that we want to examine. It can be the orientation of events, orientation of cell division. It can be the volume of the cells. It can be the area of the cells. It can be changes, dynamic change, whatever you want. Now it's relatively simple to quantify this from the segmented data and just to map them, which is, of course, the basis for a lot of spatial statistics works that we're trying to do now. Okay.
This is just one point in time.
No. So it depends on which one. So this, for example, the color coding is for the time.
That's the time encoding right there.
Right, and here
Right, so that's some kind of movie.
That's based on the movie, right?
Yeah.
And also this is based on the movie. This is like when the cell is invaginating into the inside of the embryo. So some of them are you can do either this or that. You can do a spatial temporal or just temporal. Right. Yeah. Okay, so single cell activities are usually relatively easy to identify inside the embryo, but many times you want to study cellular behaviors that are much more complex than that. For example, one of the most studied processes in Drosophila development is germinal extension or axis elongation. All organisms more or less have this one primary axis, even as humans, and the elongation process is something that we've been studying in multiple organisms. Here it's called germinal extension. You can see it takes about 45 minutes or a bit less. And one of the the key driver of this elongation, it's called rosettes or multicellular multicellular intercalations. The simplest form is when you have 4 cells. You cannot do that with less than that. And what you can see over time is that the right and the left cells are gradually losing the interface between them until the formation of that rosette in the middle, and then a new interface is formed between the top and the bottom cells. And if you zoom out from this entire thing, you can see that it starts from being vertically elongated to horizontally elongated. And when you do this thing many times in the same tissue, same orientation, then the entire tissue is undergoing convergence and extension. This is the main driver of this process. And you can do that with 5 cells, 6 cells, as you can see here, and up to, we saw even 11 cells. So rosettes have been studied for a very, very long time, and most of the papers have been a bit stuck with modeling these rosettes because it's very difficult to extract them from the data. And so there have been some algorithms that identify these dynamics from the data, but well, they were based on what the programmer of that lab thought a rosette is, which is one problem we saw, and each one of these algorithms was targeting a single cell activity that they were interested in. We wanted something that will be for any cell activity, and that will be based on what I think a rosette is, not what their programmer thinks. And so the trick here is the following. So it starts with the user just labeling one example of what the activity looks like. For example, this is a 4-cell rosette that the user is interested in finding, providing that to the algorithm. The algorithm converts that into a multi-dimensional time sequence. In this case, every dimension is the distance in microns between one pair of cells, and choose 2 distances. And so the distance between the 2 red ones is this curve. The distance between the 2 green ones is this curve. And where they meet here is exactly where you have the intercalation. Right. So now, after that, the algorithm will convert all the quartets in the embryo into time series. And now we have a multi-dimensional time series search problem, which is something that you guys do sequence analysis. It's a 30-year-old problem, 40-year-old problem. Okay, so one way to solve that is simply by a sliding window, just to search Euclidean distance between the 2 sequences. But the problem is that it's not exactly like DNA. I mean, like rosettes can be slower, they can be faster, they can be first slow and then fast. We need something that would allow time to stretch when we're matching this pattern. And so we used not a new trick, but actually the oldest trick in the book, which is dynamic time warping. So this algorithm was actually the first algorithm for pairwise sequence alignment. Allows us to match the 2 sequences. And the novelty was that it allowed us to have gaps or deletions in the sequences so that we can locally stretch and compress the sequences to match them perfectly to one another. And we use the same trick exactly, but not on ACTG, it's simply on multi-dimensional numeric array. And as you can see here, the match was done perfectly, and indeed we have here a rosette. So that allows us that opens for us the ability to map more or less any cell activity or multicellular activity that we want in the data. Of course, this data is now being used in my lab for multiple studies of mechanism of how development is working, but we're focusing on tools here, so I'm going to continue to the next one. Okay, so we published the paper on the whole embryo segmentation around mid-22, I think. In 2023, we didn't see it echoing much. I mean, like there wasn't a lot of noise about it. And then since the beginning of 24, every few months, we saw another lab that just downloaded the one single, and these are all the same embryos, the one embryo that you saw now, downloaded and published an entire paper on that, and each one of them is like a completely new idea. So UC Santa Barbara developed like the most detailed biophysical model on how axis elongation is happening. Princeton used that in order to develop an algorithm for local strain mapping, local deformations of the embryo. From MIT, they used that in order to develop a GNN that will learn what the cell is going to do next and where the folds are going to happen. Columbia used that in order to identify dynamic patterns novel dynamic patterns in selectivities. Also, our own George is currently working on developing a model that shows what's the differences in the contribution of different rosette sizes. So with each one of these, we realize two things. Well, first of all, there is really an urgent need. I mean, like the community is ready to analyze this data. I mean, like people know what they want to do with that. And so we need to somehow make that more accessible, this algorithm. But the second thing is that knowing most of these labs, I can tell you, they are uber computational. I mean, like they have very experienced programmers in their labs. And that was kind of it was nice, but it was also a bad thing because it means that we're not accessible to the huge experimental community of Drosophilists that I mean, like most Drosophilists are experimental, and we want them to enjoy what we have to offer here. And so the question is, how can you integrate that into an experimental research? And so this is the basis for motivation for the last thing I'll show you today. So, second.
I have a personal
Yeah.
question. So you are studying one embryo, right?
Oh, we're using multiple embryos in every study, but yeah.
So is there any way to compare different embryos or a kind of perturbation study?
Yeah.
Do you have any plans?
Well, yeah, we're working. Well, the simplest way is, you know, you have mappings, right? You just look at the maps of this and that. So that's already something, right? The mapping of the activities that you saw before in the atlas. Just by looking at them, you'll see differences. I mean, these mappings are extremely sensitive. You will not find any mutation in which you will not clearly see the impact. But apart from that, of course, you can align the embryos here in point drift or whatever, and
Okay.
have quantitative comparison. Yeah, it's a okay. So the guy to see here is Matej Kreins. He's a biophysicist, is an expert in vertex models, which is a model that we use to simulate tissue dynamics. He's working in Slovenia in the Joseph Stefan Institute, and together with him, we're currently working on a resource project, let's say. So what you see here is called GRAPE Graph Embryo Project, and it's an online wearable database for whole embryo, whole fly embryo analysis. So you go online, you don't need to pay, you don't need to register, you don't need to be in a university, like whatever, wherever you are, just go online. And what you see here is this. So you can ask more or less any query that you want about the embryo. Give me the first 50 cells that divide. Give me all the interfaces that can completely contract in the posterior end of the embryo. Whatever it is that can be extracted from the embryo, you can ask him. Your query will be sent to Slovenia to a server. So you don't need to worry about your resources. And after a few seconds or a few minutes, it will come back to you and be shown in the form of this three-dimensional embryo. Here in this example, the query, the result was the person was interested in these cells here that are just about to invaginate. All right. So of course you can rotate the embryo, you can save a JPEG, you can use it for a paper. It's all, you know, it's available for you. If you have numeric results, if you ask like for numbers of cell areas or whatever, you can also click to ask for an Excel sheet to be downloaded, take it to your computer, do a t-test, whatever you want. Now, as for the language of the queries, so as we said before, some of these queries can take, at least in MATLAB, like 100, 200, 300 lines to program, even more. That's not suitable if you're not from computer sciences. But what Matej has really evidently identified is that our tissue, very much like any tissue in biology, can be represented as a knowledge graph. So in case you're not familiar with that, knowledge graphs are one of the most powerful constructs in graph theory. It basically is the way to represent the relations between different entities. For example, patient birthday, and then sensitivity to drugs, and then what was the symptoms. And all of these things which are conceptually different, a knowledge graph will allow you to associate them and to point to the different connection between them. And that's the same idea here. So we define an issue in terms of the tricellular vertices between the cells, the interfaces between two cells, the cells themselves, and then the time point. And all of this creates an absurdly compact way to query the data, and it's fully generic. For example, the query that brought, I think it was the query that brought that, was written in like 4 or 5 lines that are very simple and very intuitive. So that's already like one big step toward like simplicity. If you're not familiar with this language, then we're giving you here like 10 or 15 sample queries that are fully annotated. You can look at them, you can learn from them, you can copy paste them and then change them to what you want, and learn from that. And to try to even make it simpler, we're currently considering we're checking if it's possible to integrate a GPT that will take from you whatever you want in human language and translate that into the query language, and then do that for us. So it will be absolutely breaking the wall between non-computational and computational labs. Okay, so I think, yeah, that would be a good time to finish. I just want to thank everyone in my lab and my postdoc advisors for supporting this research. Of course, thank you, Marcy, for the invitation. Right. And I'll be happy to hear some questions.
A long way.
I'm curious about the segmentation algorithm.
Yeah.
So you are using watershed, but I heard that when using segmentation algorithm, there is an issue. So that's why there are a lot of other alternatives, something like depth algorithm. So do you have any issue when studying Embryo about the poor accuracy of the segmentation algorithm? I mean, it's not working well, something like that.
Okay, so look, in general, they're saying there are problems. Segmentation tracking is a field. It's becoming a field. Like every week on ResearchGate, you see like 5 new papers. Every 4 months, a big new paper. So people are really, it's a really hot thing now. So if there are problems, like as much as you have in sequence analysis, watershed is a very old technique, but it's perfectly suited for cell membrane information because that's the definition. You're looking for the watershed, which is the membrane between them. So for 2-dimensional data, it would work perfectly. If it would work in 3-dimensional data, the answer is absolutely no. Not only because of inaccuracy, but also because the membrane floor for is only lateral in these stages. It's not in the apical side. So the water will just spill out. So you cannot do more than that. And I was asked quite a lot, like, you have units, why are you using watershed? Like, because it worked the best. I don't feel any strong emotions to new technology. Now we Ria spending her postdoc on, we're trying to recover the full cellular tree of the flight to get to like hundreds of time points with 99% maybe success. And she's of course deep learning, networks, like so if you want something so challenging, you need to move on to like deep approaches. But for this one, like it just worked best.
I guess, the big breakthrough with segmentation, especially in 3 dimensions, was the UUNet and, deep neural nets, right? And ImageNet and that kind of thing. So I was wondering how that fit into your thinking.
Right. So, today, CellPose, Ultrac, all of these are doing fantastic work. But when I say fantastic, I mean, it's amazing compared to what people did before, but it's really still far from having like single cell resolution accuracy. What we do is we're not trying to compete with them in this area. We're saying like we're developing an algorithm that automatically identifies. We take CellPose specifically, the results of CellPose, and we developed an algorithm that is identifying errors and automatically corrects the errors of CellPose in segmentation and tracking. So we're kind of generating a boss that looks over the work and
yeah, I mean, hybrid approaches. I mean, where you utilize these different approaches to help you kind of zoom in on better structure that you can measure with some kind of figure of error. That makes a bunch of sense.
Right. We are solving a few problems with the unit, but we're not trying to develop the core modules that do that. We're trying to integrate them into one way.
Just another one. I mean, what biological problem? I mean, so this is great methods and exciting times for the field. I can see that, and having the tool that allows other people's access, especially if you're pulling, going to Slovenia to get the cycles. So, but what is your motivation in terms of biology? What are the key questions that you want to answer?
Right. So first of all, the gastrulation is such a complex morphogenetic process. We have multiple projects, and one of them is, as I said, to understand the role of multicellular rearrangements. There are many works on that, but some of them, almost all of them, try to ignore the role of Rosette. So this is one medium-sized project, and the bigger one is that we're trying to, shall I say, reconstruct the dictionary of all the repeating cell activities that guide the genetic process, and then we're currently working on how to associate it with very deep sequencing, like gene expression data, so we could systematically understand how different organs are formed, what are the differences in terms of the cell activities that they utilize in order to form the different morphologies. Basically, we're trying to understand how different organ morphologies are forming.
Mhm. Thank you.
Yeah, so you just if I understand correctly, you just said like the other segmentation methods like cell pulls, like they're not doing very well on segmenting like the software embryos.
No, it's doing phenomenal work. I think that Cepos is hands down probably surely one of the 3 top algorithms, but it still accumulates error very fast. I mean, you have 5,000-6,000 cells every time point, and it's increasing all the time. And when the cells go in, the fluorophore level is going down. So it's a bit much to expect a pure segmentation algorithm to manage all of this. And Cepos is doing phenomenal work. It's still far from what we need.
There isn't like a uniform like software that people use for in this case, and also like segmenting like the software embryos is difficult compared to segmenting like mice or human cells. Like the cells in the mouse embryo, they kind of like stack into each other really dense.
Right, yeah. So you have advantages and disadvantages. So Drosophila are easier to handle. We know how to image them. We know how to fix them inside the agros, and the more challenging part in them is that they have yolk, for example, and yolk is autofluorescing. So you saw that there is like a white smear in the middle. That's a huge problem. And we're trying to solve. So each one of, and in humans you don't have that. And you start from a smaller number of cells in mice and humans, obviously. You start from a single cell in the fly. When you cellularize, you start with 5,500 cells.
Yeah.
So it's
exactly.
But there I think that it's more difficult to keep them alive, and I don't know exactly how they do that, but I think that they have other problems there.
Yeah,
it really depends on the model. Yeah.
I'm also familiar with like microscopy, but regarding this method we use with the light sheets, is like photosensitivity or phototoxicity ever a concern, or is that not right?
It's exactly the opposite. It's absurdly low phototoxicity. I'm imaging flies for the entire day of their embryonic development, and they hatch.
Oh.
So the way it works is like with a point scanner, you're shining an entire beam, you're shining an entire line inside the tissue, but you're collecting only the dot in the focal plane. So you're really exhausting a lot of morpho here. Light sheet, you're shining 2-dimensional plane, and you're collecting the 2-dimensional plane in a matter of like 15 milliseconds. So it's one of the biggest advantages of light sheet microscopy. Yeah. Okay. Thank you.